Have you ever wondered why you have your mum's eyes or your dad's smile? This big question about how things are passed down from parents to children fascinated a young man named Gregor Mendel long ago. He was born in 1822 in a small village in what is now the Czech Republic. His family were farmers, and from a very young age, Gregor was surrounded by plants and animals. He spent his days in the family orchard helping his father graft fruit trees. He learned how to care for the gardens and saw firsthand how different plants could have different features. This early life on the farm planted a tiny seed of curiosity in his mind, a seed that would one day grow into a huge scientific discovery. Even as a boy, Gregor was a brilliant student. He loved learning, especially about the natural world. His family saw his potential and wanted him to have a better education than they did. Life was not always easy, and they made many sacrifices to send him to school. He studied hard and excelled in subjects like physics, mathematics, and science. His teachers were amazed by his bright mind and his passion for understanding how things work. This strong foundation in science and math was incredibly important. It gave him the special tools he would need later to solve one of nature's greatest puzzles, a mystery that no one else had been able to figure out before him. His journey into science was not a straight path. To continue his education without placing a heavy financial burden on his family, Mendel made a life-changing decision. At the age of 21, he joined the Augustinian Abbey of St. Thomas in Brno. Becoming a monk might seem like an unusual choice for a budding scientist, but this monastery was a vibrant center of learning and scientific inquiry. It was a place where faith and reason lived side by side. The abbey had an extensive library, botanical gardens, and a community of scholars who were interested in everything from astronomy to agriculture. This environment was the perfect place for a curious mind like Mendel's to flourish and grow. Living in the monastery gave Mendel the freedom to pursue his intellectual passions. He was not just a monk. He was also a teacher and a researcher. He taught physics and natural science at a local school, where he was known for being a kind and patient instructor. But in his quiet moments, his mind always returned to the fields of his childhood and that one big question: how are traits passed down? He saw it in the flowers, in the monastery garden, and in the crops grown nearby. He knew there had to be a pattern, a set of rules that nature followed. He was determined to find them, using his unique background in both farming and formal science. Mendel's interest in heredity wasn't just a passing thought. It was a deep and persistent puzzle he felt compelled to solve. At the time, most people believed in an idea called blending inheritance. They thought that traits from a mother and a father simply mixed together in their offspring, like mixing black and white paint to get grey. But Mendel, with his keen eye for detail, saw problems with this theory. He noticed that traits didn't always blend. Sometimes a grandchild would have a feature like red hair that their parents didn't have, but a grandparent did. The traits seemed to disappear and then reappear. This told him that something more complex and more interesting was happening. His formal training made him the perfect person to investigate this mystery, and after joining the monastery, he was sent to the University of Vienna to study science and mathematics in greater depth. There he learned from some of the best scientific minds of his time. He studied plant anatomy, physics, and most importantly statistics and probability. This mathematical training was his secret weapon. While other naturalists were content to simply describe what they saw, Mendel learned how to use numbers to find hidden patterns. He realized that to truly understand heredity, he couldn't just observe, he had to count, measure, and analyze. Back at the monastery in Brno, Mendel was given a small plot of land in the garden to conduct his experiments. This garden became his outdoor laboratory. It was here that he would spend the next 7 years, from 1856 to 1863, meticulously working with tens of thousands of pea plants. Imagine the patience this required. He wasn't just planting seeds and waiting to see what would grow. He was acting like a detective, carefully controlling every step of the process. He chose the common garden pea plant, Pisum sativum, because it was easy to grow, had many different varieties, and he could control its pollination, which was key to a successful experiment. His life as a monk provided the ideal setting for this long and detailed work. The monastery offered a quiet, stable environment where he could focus without the distractions of the outside world. He had the time, the space, and the intellectual support of his fellow monks. His days were structured around prayer, teaching, and his scientific research. This unique combination of a farmer's practical knowledge, a scientist's formal training, and a monk's patient dedication allowed him to design and carry out an experiment that was far ahead of its time. He was ready to ask nature a question and listen carefully for its answer. The success of any great experiment often depends on choosing the right subject to study. And Mendel made a brilliant choice, the pea plant. This wasn't a random decision. He needed a plant that had specific characteristics to help him uncover the rules of inheritance. First, pea plants grow quickly and produce many offspring in a single generation, which meant he could gather a lot of data in a relatively short amount of time. He didn't have to wait years to see the results of his crosses. This allowed him to repeat his experiments over and over again to make sure his findings were reliable and not just a fluke. Second, pea plants have a number of traits that come in simple, easy-to-see, either-or forms. Mendel carefully selected 7 of these traits to study: flower color, which could be purple or white; seed shape, round or wrinkle; seed color, yellow or green; pod shape, inflated or constricted; pod color, green or yellow; flower position, axial or terminal; and plant height, tall or short. There were no in-between options for these specific traits. A plant was either tall or short, not medium-sized. This clarity was crucial. It allowed him to track each trait without confusion, which would have been impossible if the traits blended together into a fuzzy middle ground. Another key advantage of pea plants is their flower structure. Pea flowers naturally self-pollinate, meaning the pollen from a flower fertilizes the ovules of the same flower. This created a perfect control group for Mendel. He could let a plant self-pollinate to produce what he called true breeding lines, plants that, when self-pollinated, would always produce offspring with the same trait. For example, a true breeding tall plant would only ever produce other tall plants. This gave him a stable starting point for his experiments, ensuring that the traits he was observing were consistent and pure. But the real genius of his setup was that he could also easily cross-pollinate the plants by hand. Mendel could play the role of a busy bee, carefully transferring pollen from one plant to another with a small paintbrush. He would open a flower bud, remove the male parts, the stamens, to prevent self-pollination, and then dust the female part, the pistil, with pollen from a different plant he had chosen. This painstaking process allowed him to control exactly which plants were mating. He could cross a tall plant with a short plant, or a purple-flowered plant with a white-flowered one, and then observe the traits of their offspring with complete certainty about their parentage. What made Mendel's work truly revolutionary was not just what he studied, but how he studied it. He brought a new level of precision and mathematical thinking to the field of biology. Before Mendel, many naturalists studied inheritance by looking at the whole organism and all its complex traits at once. This was often confusing and led to messy, inconclusive results. Mendel's brilliant idea was to simplify. He focused on a single clear trait at a time, like flower color. By isolating one variable, he could track it carefully across generations without getting distracted by other characteristics. This methodical approach was a game-changer for biological experiments. Mendel was also a pioneer in the use of controlled experiments. He didn't just plant seeds and see what happened. He designed each step with a specific question in mind. His first step was always to create true breeding parent plants, which he called the P generation, for parental. He did this by letting plants self-pollinate for several generations until they consistently produced offspring with only one form of a trait. Once he had his pure lines, for example, a line of only purple-flowered plants and a line of only white-flowered plants, he would then cross-pollinate them. This first generation of offspring he called the F1 generation, for first filial, from the Latin word for son. His innovation didn't stop there. After observing the F1 generation, he would then let these plants self-pollinate to produce the next generation, which he called the F2 generation. This multi-generational approach was essential for seeing the hidden patterns of inheritance, but perhaps his most important method was his insistence on using statistics. Mendel didn't just say, some of the offspring had purple flowers and some had white. He counted every single plant. He recorded the exact numbers of offspring with each trait. Over his 7 years of experiments, he cultivated and tested nearly 28,000 pea plants. This large sample size was vital for seeing the statistical patterns emerge from the data. Keeping detailed and organized records was the final piece of his scientific puzzle. For every single cross he made, Mendel meticulously documented the parents, the offspring, and their traits. His notebooks were filled with numbers and careful observations. This rigorous record-keeping allowed him to analyze his results and discover the mathematical ratios that were at the heart of heredity. He wasn't just gardening, he was collecting data. This combination of focusing on single traits, using controlled crosses, applying mathematics, and keeping immaculate records set a new standard for science. He was applying the methods of physics and chemistry to the study of life. Through his careful counting and analysis, Mendel discovered fundamental rules that govern how traits are passed down. The first of these is now known as the law of segregation. When he crossed his true-breeding purple-flowered plants with his true-breeding white-flowered plants, he found something amazing. All of the offspring in the first generation had purple flowers. The white flower trait seemed to have vanished completely. This directly challenged the idea of blending inheritance. The flowers weren't a pale lavender, they were all a vibrant purple. He called the purple trait dominant and the white trait recessive because it seemed to recede into the background. The real magic happened in the next step. When Mendel allowed the first generation of purple-flowered plants to self-pollinate, the lost white flower trait reappeared in the second generation. He counted the plants and found something remarkable. For every 3 purple-flowered plants, there was approximately 1 white-flowered plant. This 3-to-1 ratio appeared again and again in his experiments for all 7 traits he studied. From this, Mendel reasoned that each plant must carry 2 factors, which we now call genes, for each trait, one inherited from each parent. He concluded that these 2 factors separate or segregate from each other when sex cells like pollen and ovules are formed, and each sex cell receives only 1 factor. Mendel then took his experiments a step further to see if different traits were inherited together or separately. For example, did a plant that was tall also have to have purple flowers? To answer this, he performed dihybrid crosses where he tracked 2 traits at once. He crossed a plant that was true breeding for round yellow seeds with one that was true breeding for wrinkled green seeds. As expected, the first generation showed only the dominant traits. All the plants had round yellow seeds. But when this first generation self-pollinated, the second generation offspring showed a fascinating mix of traits. He found not only the original parental combinations, round yellow and wrinkled green, but also 2 new combinations, round green and wrinkled yellow. When he counted all the different types of seeds, he found a consistent ratio of 9-to-3 to 3-to-1. This led to his second great discovery, the law of independent assortment. This law states that the factors for different traits are sorted into sex cells independently of one another. In other words, the trait for seed shape, round or wrinkled, is inherited separately from the trait for seed color, yellow or green. It's sort of like shuffling 2 separate decks of cards instead of 1. Each trait gets passed down as an independent event. Mendel's findings were truly groundbreaking because they directly contradicted the prevailing scientific theory of the time, which was called blending inheritance. For centuries, people who bred plants and animals just assumed that offspring were simply a diluted mix of their parents' characteristics. If you crossed a tall plant with a short plant, you would expect to get a medium-sized plant. This idea seemed logical on the surface, kind of like mixing 2 colors of paint. It explained some observations, but it couldn't account for many others, like why traits sometimes skip a generation and reappear later, looking exactly as they did in the grandparents. Mendel's experiments provided a much better explanation. His results showed that inheritance is not about blending. It's actually about discrete individual units or factors. These factors, which we now call genes, remain separate and distinct from one generation to the next. When Mendel crossed his tall and short pea plants, the F1 generation were all tall. The factor for shortness didn't get blended away or diluted. It was just hidden or recessive. It was still present in the F1 plants, ready to be passed on. This concept of dominant and recessive traits was completely new and explained how a characteristic could vanish and then reappear unchanged in a later generation. The reappearance of the short plants in the F2 generation was really the final blow to the blending theory. According to the blending idea, once the tall and short traits had mixed to create the F1 generation, they should never be able to unmix. You can't take grey paint and separate it back into pure black and pure white, but Mendel showed that this is exactly what happens in genetics. The short plant trait reappeared in the F2 generation in its original pure form, not as a slightly less tall version. This demonstrated that the hereditary units are passed down whole and intact, like little marbles of information, not like liquids that can be blended together. This particulate theory of inheritance was a radical shift in thinking. It proposed that heredity worked by a set of predictable rules and could be understood through mathematics. Mendel's factors were not vague, mysterious fluids, but concrete entities that could be counted and tracked. This idea laid the groundwork for the entire field of genetics. It meant that inheritance wasn't a messy, unpredictable process, but an elegant system of information being passed from parent to child according to clear, logical principles. He replaced a fuzzy, incorrect idea with a sharp, clear and correct one, even if the world wasn't quite ready to hear it yet. While Mendel's laws were a monumental breakthrough, they were really just the beginning of our understanding of genetics. The world of heredity is even more wonderfully complex than what he observed in his pea plants. Scientists who followed in his footsteps discovered new patterns of inheritance that didn't follow the simple dominant and recessive rule. One of these is called incomplete dominance. This is when the heterozygous offspring, meaning having one factor for each trait, shows a phenotype that is a blend of the two parental traits. For example, when you cross a red snapdragon flower with a white one, you don't get red flowers, you get pink ones. This kind of looks like blending, but it's not. The red and white factors are still separate, and they can reappear in later generations. Another fascinating pattern is codominance. In this case, both alleles, different versions of a gene, are expressed equally and fully in the offspring. A great example of this is the ABO blood group system in humans. The alleles for type A blood and type B blood are codominant. If a person inherits one A allele and one B allele, they don't get a blend of the two. Instead, they have type AB blood, where both A and B characteristics are present. This shows that the relationship between alleles isn't always a simple case of one dominating the other. Sometimes they can share the spotlight. Scientists also discovered that Mendel's law of independent assortment doesn't always hold true. It works perfectly for genes located on different chromosomes, but genes that are located close together on the same chromosome tend to be inherited together. This is called gene linkage. Think of it like beads on a string. If two beads are very close to each other, they are more likely to be passed on as a pair. This discovery helped scientists begin to map the locations of genes on chromosomes, creating a sort of roadmap of our genetic code. It added a new layer of complexity to Mendel's simple, elegant model. All of these later discoveries, incomplete dominance, codominance, gene linkage, and many other non-Mendelian patterns might seem like they contradict Mendel's work, but really, they build upon it. Mendel provided the essential foundation, the core principles of particular inheritance, segregation, and assortment. Subsequent generations of scientists have added to and refined his work, showing how these basic rules can be modified and can interact in more complex ways. Mendel gave us the alphabet of genetics. Later scientists have used that alphabet to write increasingly complex and beautiful stories about how life works. His ideas were not the final word, but they were the essential first word. One of the most remarkable aspects of Gregor Mendel's work was his deep commitment to good scientific practice, especially his use of large sample sizes. He didn't just grow a handful of pea plants, he grew thousands upon thousands of them over many years. When he studied the F2 generation for a single trait, he often counted over 8,000 individual plants. Why was this so important? Because in the world of biology and probability, patterns only become clear with large numbers. If he had only planted 10 seeds, he might have gotten 7 purple flowers and 3 white ones, or maybe even 6 and 4. The results could have been dismissed as random chance. By using such a large sample, Mendel was able to see the true mathematical ratios emerge from the noise of random variation. The consistent 3-to-1 ratio he observed wasn't just a coincidence. It was a statistically significant result that pointed to an underlying biological law. This application of quantitative analysis to a biological problem was revolutionary for his time. Most biologists were naturalists who focused on describing and classifying organisms. Mendel, with his background in physics and mathematics, approached the problem like a statistician, letting the numbers really tell the story. His patience and dedication to collecting vast amounts of data were just as important as his brilliant insights. His scientific method was impeccable in other ways too. He was incredibly methodical in his approach, starting with the creation of true breeding lines to make sure he had a pure and reliable starting point. This control over his initial variables was critical. He then focused on only 1 or 2 traits at a time, which simplified the problem and allowed him to isolate the effects he was studying. This careful step-by-step process prevented the confusion that had plagued earlier attempts to understand heredity. He designed his experiments to ask clear questions and get clear answers, eliminating as many outside influences as possible. Furthermore, Mendel's integrity as a scientist is shown by the fact that he repeated his experiments for 7 different traits and found that the same mathematical patterns held true for all of them. This repetition confirmed his results and gave him confidence that he had discovered a universal principle of inheritance, not just a quirk of pea plant flower color. His combination of a large-scale, careful controls, mathematical analysis and rigorous repetition really set a new gold standard for biological research. He demonstrated that the living world, just like the physical world of planets and atoms, operated according to predictable and understandable laws. Despite the brilliance and importance of his work, when Gregor Mendel published his findings in 1866 in a paper called Experiments in Plant Hybridization, the world barely noticed. His paper was sent to libraries across Europe, and he even sent a copy to Carl von Nageli, a leading botanist of the time. Unfortunately, the scientific community was just not ready for his ideas. His work was a strange mix of botany and mathematics, 2 fields that were rarely brought together. Most biologists at the time did not have the mathematical training to understand the significance of his ratios. They were focused on Darwin's new theory of evolution and were looking for gradual, continuous changes, not the discrete mathematical jumps that Mendel described. For over 3 decades, Mendel's groundbreaking paper sat on library shelves, gathering dust. He continued his duties at the monastery, eventually becoming its abbot, which left him with less time for his scientific pursuits. He passed away in 1884, likely believing that his years of painstaking work with pea plants had been forgotten and had made no impact on the world of science. It is a poignant part of his story that the man who laid the foundation for modern genetics died without ever knowing the true importance of his discovery. His work remained a hidden secret, waiting for the right moment to be unearthed. That moment finally came in the year 1900, 16 years after his death. In a remarkable coincidence, 3 different scientists from 3 different countries, Hugo de Vries, Karl Correns and Erich von Tschermak, independently conducted their own experiments on inheritance. As they prepared to publish their findings, they each did a search of the existing scientific literature, and one by one, they all stumbled upon a long-forgotten paper by an Augustinian monk named Gregor Mendel. They were stunned to find that he had not only reached the same conclusions they had, but had done so more than 30 years earlier with incredible precision and clarity. The rediscovery of Mendel's work was like a spark that ignited the field of biology. The scientific world was now ready for his ideas. The study of cells and chromosomes had advanced, providing a physical basis for Mendel's abstract factors. Scientists quickly realized that his laws of inheritance were the missing piece of the puzzle needed to understand both heredity and Darwin's theory of evolution. They gave his principles the name Mendelian genetics, and he was posthumously given the title he so richly deserved, the father of modern genetics. His story is a powerful lesson that a great idea can sometimes be ahead of its time, but its truth will eventually shine through.